nobody expected the comparison to come back at 90%. When the team that sequenced the domestic cat genome placed their results alongside the human genome and ran the similarity analysis, the figure that emerged was higher than most researchers had predicted going in. Roughly 90% of the functional DNA in a house cat has a direct equivalent in the human genome. Genes that do the same job, built from recognizably similar code, producing proteins that operate the same biological machinery. The number surprised the researchers who found it, and it surprised the wider scientific community when the results were published. It shouldn't have, in retrospect, but it did, because nobody had sat down and done the full comparison before. The comparison was possible because of a cat named Cinnamon. In 2007, a team at the National Institutes of Health sequenced the complete genome of an Abyssinian cat living in Missouri. Cinnamon became the first domestic cat to have her full genome read, and the sequence that came out of that project became the reference against which everything else was measured. The choice of Abyssinian wasn't random. Abyssinians have a relatively uniform genetic background, which makes sequencing cleaner and the results easier to interpret. Cinnamon's genome ran to about 2.7 billion base pairs, somewhat smaller than the human genome's 3 billion, and it contained roughly 20,000 protein-coding genes, similar to the human count. The sequencing project took years. The comparison against the human genome took considerably less time once the cat sequence was complete. A percentage like 90% sounds straightforward until you ask what the other 10% contains. The 10% that differs between cat and human DNA is not distributed randomly across the genome. It clusters in specific functional categories, mostly related to the aspects of biology that have diverged most between species that split from a common ancestor roughly 80 million years ago. That shared ancestor was a small, insectivorous mammal that lived in the shadow of the dinosaurs, and both the cat lineage and the human lineage have been evolving independently ever since the dinosaur extinction cleared the ecological space for mammals to diversify. 80 million years of separate evolution should, in theory, produce two very different genomes. The reason it didn't, across most of the functional genome, is that most of what a mammal needs to do at the cellular level is the same whether it's a cat or a human. And the genes that handle those basic functions don't change much because changing them would break the machinery. The scale of that shared ancestry becomes clearer when you compare it to other familiar numbers. Humans and chimpanzees share around 98.7% of their DNA. Humans and cats share 90%. Humans and mice share roughly 85%. The gap between cats and mice might seem small in percentage terms, but in a genome of billions of base pairs, those 5 percentage points represent tens of millions of individual differences in the code. More importantly, the differences are not evenly distributed. The extra 5% similarity between cats and humans relative to mice tends to fall in the functional regions of the genome, the parts that actually get used to build proteins and run biological systems. It's not that cats and humans are more closely related than cats and mice in the evolutionary sense. Cats and mice split from their common ancestor at roughly the same time that cats and humans did. The closer genomic similarity between cats and humans is about which biological solutions each lineage kept and which it modified. The shared 90% covers an enormous range of biological functions: genes for cell division, energy metabolism, immune response, tissue repair, hormone production, and the construction and maintenance of the nervous system all appear in both genomes in highly similar forms. The proteins these genes produce are often interchangeable between species. Some drugs developed for human conditions work in cats because the target proteins are similar enough. Some pathogens that affect humans can affect cats for the same reason. The biological systems that the shared DNA builds are, in most of their essential features, the same system running in a different body. The 90% figure is not a coincidence or a curiosity. It reflects the fact that mammals share a body plan, and building and maintaining that body plan requires a core set of genetic instructions that have remained largely intact across 80 million years. For most of the 20th century, the mouse was the standard model for human biology in medical research. Mice breed quickly, are easy to keep in laboratory conditions, and their genomes had been studied extensively. When researchers needed to test how a disease progresses or how a drug works at the genetic level, mice were the default choice. The problem that accumulated over decades was that mouse results often didn't translate to human patients. Treatments that worked in mice failed in human trials at a rate that became a serious concern in pharmaceutical research. The genome comparison suggested a structural reason for some of those failures. Mice and humans share roughly 85% of their functional DNA, which is substantial. But cats and humans share 90%, and the 5% difference falls in categories that matter for disease modeling. Specifically, the cat genome shows closer similarity to the human genome in the regions connected to immune function, certain metabolic pathways, and the genetic architecture of several major diseases. These are precisely the categories where mouse models most often fail to predict human outcomes. The comparison didn't mean mice were useless as research models. They remain valuable for many purposes, but it raised a question that researchers hadn't asked clearly before. If you're trying to model a human disease at the genetic level, which animal's genome is actually closest to the human version of that disease? For a growing list of conditions, the answer turned out to be the domestic cat. Cats develop diabetes in a way that is genetically almost identical to how type 2 diabetes develops in humans. The same genes are involved, the same cellular mechanisms fail, the same patterns of insulin resistance appear. Cats on high carbohydrate diets develop the condition at rates that mirror human populations on similar diets, and the progression of the disease in cats follows the same stages as the human version. For decades, veterinarians and endocrinologists had noticed the clinical similarity without being able to explain it at the genetic level. The genome comparison provided the explanation. The genes governing glucose metabolism and insulin signaling are among the most conserved between cats and humans, sitting in the 90% shared region of the genome. When those genes malfunction in a cat, they malfunction the same way they do in a human, because they are, in most meaningful respects, the same genes. Kidney disease in cats follows a similar pattern. Chronic kidney disease affects a very high proportion of older cats, and the progression of the disease, the way the kidneys lose function over time, the pattern of protein loss, the secondary effects on blood pressure and red blood cell production, all of it mirrors how the condition progresses in elderly humans. The veterinary literature on feline kidney disease and the medical literature on human kidney disease have been drawing on each other for years, and the genome comparison gave a genetic basis for why that cross-referencing was valid. The kidney function genes in cats and humans are closely matched. What damages them is similar. How they fail is similar. The comparison confirmed that studying kidney disease in cats and studying it in humans were never as separate as the different medical specialties implied. When researchers compared feline cancer genetics to human cancer genetics, the overlap was substantial enough to change how some research programs were being designed. Certain cancers in cats, particularly mammary tumors and some lymphomas, are driven by the same genetic mutations that drive equivalent cancers in humans. The tumor suppressor genes that fail in human breast cancer are present in cats in similar form, and when they fail in cats, they fail in similar ways. The gene that is most commonly mutated in human cancers, a gene called p53 that regulates cell division and prevents uncontrolled growth, is present in the cat genome in a version so similar to the human version that the protein it produces functions almost identically. That similarity means that a cat with a tumor driven by a p53 mutation is, at the genetic level, experiencing a disease whose mechanics are very close to the equivalent human cancer. The practical consequence of the cancer findings was that certain drugs under development for human cancers began being tested in cats not because cats were convenient, but because the genetic target was similar enough that a positive result in a cat would be more informative than a positive result in a mouse. A small number of veterinary oncology programs and human oncology programs began sharing data in ways they hadn't done before because the genome comparison had demonstrated that the data was comparable. This wasn't framed as cats replacing mice in research. It was framed as using the genetic similarity that the comparison revealed to make research more accurate for specific disease categories. The genes that build and maintain the nervous system are among the most conserved across all mammals, and the cat-human comparison showed that clearly. The basic architecture of the mammalian brain, the types of neurons, the signaling chemicals they use, the way different brain regions communicate, all of it is built from a set of genes that appear in almost identical form in cats and humans. The structural similarity between a cat brain and a human brain goes well beyond what you'd expect just from the fact that both are mammals. Cats show sleep patterns that closely resemble human sleep patterns, including the same stages of deep sleep and the equivalent of REM sleep. The genes that regulate sleep cycles in cats are close enough to the human versions that researchers studying sleep disorders began looking at feline sleep genetics for insights that mouse models hadn't been able to provide. The parallels in sleep genetics have a practical side. Humans suffer from a range of sleep disorders, from insomnia to narcolepsy to disruptions in the deep sleep stages associated with memory consolidation. Finding genetic targets for treating those conditions requires animal models that sleep the way humans do. Mice have sleep architecture that diverges from the human pattern in several respects. Cats, whose sleep genetics sit closer to the human version, cycle through sleep stages in patterns that are more comparable to what researchers observe in human patients. Some of the first genetic markers associated with human narcolepsy were identified partly through research in cats that showed a similar condition, driven by mutations in genes that the genome comparison later confirmed were closely matched between the two species. Memory formation and learning in cats also operate on genetic machinery that closely parallels the human version. The proteins involved in strengthening neural connections during learning, the receptors that regulate how memories form and decay, the chemical signals that consolidate experience into long-term storage, all of these are encoded in genes that sit in the shared 90% of the cat-human genome. The implication is not that cats are as cognitively complex as humans. The cognitive differences are real and significant, and they show up clearly in the 10% of the genome that differs. But the underlying cellular machinery of memory and learning runs on shared components. The difference between how a cat remembers and how a human remembers is largely about scale and connectivity, not about fundamentally different genetic systems. The cat immune system is organized along lines that are remarkably similar to the human immune system, more similar than the mouse immune system is to either. The cells, the signaling molecules, the way the body identifies and responds to foreign pathogens, all of it runs on genes that appear in comparable form in both genomes. This has a direct consequence for how certain infectious diseases behave. Feline immunodeficiency virus, which affects cats, is biologically similar to HIV in humans in ways that go beyond surface resemblance. Both viruses attack the immune system using similar mechanisms, because the immune systems they attack are built from similar genetic components. Research into feline immunodeficiency has contributed to understanding HIV, and the genomic basis for that contribution is the similarity between the immune genes in cats and humans that the 2007 comparison made explicit. The 10% that differs between cats and humans is where the two species have taken separate evolutionary paths in response to different selective pressures. Cats are obligate carnivores, and that dietary specialization shows up in the genome. Cats have lost several genes that humans use to synthesize nutrients from plant sources, because those nutrients come ready-made in meat. A cat cannot make vitamin A from plant precursors the way humans can. A cat cannot synthesize taurine, an amino acid critical for heart function, because its wild ancestors ate enough meat that the gene for making it was never needed and eventually degraded. The genes that handle these functions in humans exist in the cat genome as remnants, inactive sequences that used to do something but no longer do. Cats have roughly twice as many genes dedicated to smell as humans do. The olfactory receptor gene family, which encodes the proteins that detect scent molecules, is dramatically expanded in cats compared to the human version. Humans have around 400 functional smell receptor genes. Cats have closer to 800. The difference reflects the central role that scent plays in how a cat navigates the world, identifying prey, detecting other cats, and reading the chemical signals left by other animals. In contrast, the genes for color vision in cats are reduced compared to humans. Cats see the world in fewer colors because detailed color discrimination was less important than motion detection and low light sensitivity for a nocturnal ambush predator. The 10% of the genome that differs between cats and humans is largely the record of those different ecological adaptations, the sensory trade-offs made by two lineages that went different directions after their shared ancestor disappeared. The gene for sweet taste is absent in cats. Humans have a receptor that detects sugars and produces the sensation of sweetness, which evolved to help early primates identify ripe, energy-rich fruit. Cats have no equivalent receptor because they never ate fruit, never needed to identify it, and the gene for detecting sweetness was simply never useful. The gene is present in the cat genome, but it's a pseudogene, a broken copy that produces no functional protein. Cats are genuinely unable to taste sweetness. When a cat approaches a sweet food, it's responding to the smell or the texture, not to any taste sensation. The broken sweet receptor gene is one of the clearest examples in the comparison of how the 10% that differs between cats and humans reflects 80 million years of different dietary histories. The HIV and feline immunodeficiency virus parallel is worth looking at more closely because it illustrates how the immune gene similarity translates into real research value. Feline immunodeficiency virus was discovered in 1987, and within a few years, researchers noticed structural and behavioral similarities to HIV that went beyond what you'd expect from two unrelated viruses attacking two unrelated immune systems. Both viruses belong to the same broad viral family. Both target a specific type of immune cell. Both suppress the immune response progressively over years. The reason the parallels are so close is that both viruses exploit similar weaknesses in immune systems built from similar genetic components. Research programs studying how the feline version of the virus progresses have generated findings that informed the human HIV field, not because the virus is the same, but because the immune system it attacks is, at the genetic level, built from closely matched parts. The 90% figure reframed a conversation that had been going on in medical research for decades. The conversation was about which animal models are actually useful for studying human disease. The genome comparison didn't resolve that debate because different diseases require different models, and no single answer covers all cases. But it added information that hadn't existed before. For conditions where the relevant genes fall in the shared 90% of the cat-human genome, the cat turns out to be a more accurate biological mirror of human disease than the research community had appreciated. Diabetes, kidney disease, certain cancers, some neurological conditions. All of these have a feline equivalent that runs on the same genetic machinery as the human version. The comparison made that relationship visible and gave researchers a quantitative basis for choosing models rather than defaulting to whatever had been used before. The cat sitting on a couch shares 90% of its functional DNA with the person sitting next to it. Both of them inherited that shared genome from the same small mammal that survived the end of the dinosaurs. In the 80 million years since then, the two lineages built different bodies, developed different senses, and ended up occupying completely different ecological niches. But the core of what they are, the cellular machinery that runs the heart, filters the blood, builds the nervous system, and coordinates the immune response, is built from the same set of instructions, working in nearly the same way. The comparison didn't make cats less remarkable or humans less unique. What it did was show how much of what makes a mammal work is shared across the whole group, conserved across tens of millions of years, because the biology that underlies it was solved once and kept.